told about that myself, Dr. Namita. I am working as a senior scientist in the division of floriculture and landscaping, and mainly I am working on crop improvement in rose. Rose is my crop, and in floriculture crops, rose is one of the major ornamental crop. Okay, and there are other commercial crops like chrysanthemum is there, gladiolas, marigold, orchids, anthuriums, many other commercial crops are there. But the crop improvement program, especially in India, only there are very few crops where we are working on the breeding aspects, and especially the non-conventional breeding in flower crops. And as we know that flower crops, they also plays a major role in our economy, and floriculture is just like a fashion industry. So, every day the floriculture breeders or the researchers, they are developing the varieties in flower crops. Every day on internet or any other side, we are seeing that every day for whether it is a rose, whether it is a chrysanthemum, carnation, every day we are seeing that one and another variety it is developing. And the thing is that when the color we are developing the variety with the particular trait or the novel color, then again it is constituted as a new variety. So, here my topic is what is the role of marker assisted breeding in flower crops. Okay. Before that also many they have taken the lecture on your marker assisted breeding, maybe in vegetables or in fruit crops, but since floriculture is a new area after vegetables and fruits or spices or plantation crops. So, very few breeders, they are working on the non-conventional techniques in flower crops to improve the traits. Okay. So, here just we will see the overview of the marker assisted breeding. So, we know there are different kind of markers are available, whether these are the morphological markers, cytological markers, biochemicals or DNA markers, or we can say and marker assisted breeding. Nowadays many researchers or breeders, they are mainly focused on the DNA based markers. Okay, these are some of the classifications of the markers, and mainly it is based on the whatever the PCR based markers. And if I will talk about the role of markers in floriculture crops. Okay. So, the most of the work in our country, it is based on the DNA fingerprinting or molecular characterization of the varieties, maybe in rose, chrysanthemum, carnation, orchids or anthuriums. So, whatever the varieties are these are available with us. So, the fingerprinting work it has completed in our major crops. Now, it has shifted to the development of the another kind of markers or the development of the QTLs, or these are tightly linked to the particular trait. It may be our vegetative traits or maybe our the flowering traits. Since these are the flower crops, so flowering traits are the major traits, major economic traits. So, this is only the diagrammatic representation, hypothetical diagrammatic presentations of the DNA markers. So, here for the markers which do not discriminate between the genotypes, already we know that monomorphic markers or the polymorphic markers. So, this diagram this shows the monomorphic or the polymorphic markers. So, now what is the need? What is marker assisted selection? Marker assisted selection or marker aided selection, it is an indirect selection process where a trait of interest is selected based on the markers. Marker, it may be a morphological, biochemical or DNA or RNA based markers which are tightly linked to the trait of interest. Trait of interest, it may be your flower color, it may be your fragrance, it may be your like for example in rose, maybe the density of the prickles, how many prickles are available, maybe your anthocyanin contents in rose, maybe carotenoid content in your or lutein content in marigold. Okay. So, it depends upon the what is the trait of interest. And nowadays researchers, they are also working on the different biotic and abiotic stress like many diseases. For example, if I will take the example of rose, powdery mildew resistance, black spot resistance. So, many QTLs were developed for the this biotic stress management, and similarly for abiotic stress like your drought tolerance or waterlogging tolerance in chrysanthemum. Similarly, in marigold, the QTL, the marker, it was developed for the male sterility or genetic male sterility. So, this is marker assisted selection. It is a tool which helps the plant breeder to select more efficiently for the desirable crop traits. So, why we are going for the marker assisted selections? Okay. We know when we are developing the populations, within the population we are using thousands of plants. So, phenotypically it is very difficult, or it will take more time. Till flowering we have to wait for, if we are going for any flowering trait, till the flowering is there, then that time we have to wait for that. But if we are using the different markers by this marker assisted selections, so immediately in the first year itself or F2 generation, we can use the markers, we can screen the plants populations using these markers. So, this is reducing the time, and since these markers they are not affected by the environment. So, during the emergence to the young seedling stage only you can use these markers for selection of your population for any trait. So, now what are the different prerequisites for efficient marker assisted breeding program, whether it is in, for flowers or other crops. So, first of all, it is the appropriate marker system or reliable markers. There are many kind of markers, like dominant kind of markers are there, codominant markers. So, codominant markers or recently developed markers, we have to go for those codominant markers. Okay. So, that is one of the important prerequisite. And another one is, because since we have thousands of plants in our population, so it is very difficult to go for individual plants for DNA extractions and other procedure. So, we should have a quick DNA extraction and high throughput marker selection methods. And after that, genetic maps, it is very important because it provides the framework for the marker assisted selection or marker assisted breeding. So, these linkage map, especially the high density linkage map, they are very important to go for the marker assisted breeding in flower crops. And after that, once we have a linkage maps or other genetic maps, so we should have a knowledge about the trait, marker trait associations. We have developed the markers, we have a trait, until unless we are not associating that which marker is responsible for which trait. Okay. So, that knowledge is very important before going for the marker assisted breeding. And then quick and efficient data processing. Once you know that this is a marker trait, after that you should have a tools. We should have a tools and we should have a sufficient data, so that we can immediately process that, and immediately we can find out that these are the genomic reasons. We can immediately find out this is a candidate gene. Okay, gene of the interest, and this particular marker is associated with the particular trait. So, the purpose of this slide is only to show that for the conventional breeding methods we have to wait for long times for screening of the different plants for a particular trait, but using marker assisted selections within few generations we can develop the new varieties which is having the particular trait. So, here like we are marker assisted breeding for the resistant to develop the resistant varieties like susceptible and resistant, we are developing the F1. Then upon selfing we are developing the F2 populations. And since we have a large, we have in the population we have thousands of plants. So, in F2 only we can go for the marker assisted selections using particular markers. It may be your SSR markers, it may be your SNPs, different kind of markers you can use. And in the early stage, from the emergence to the young leaf seedlings, at this stage only you will be able to select the plants which is having that particular trait. The trait, it may be for particular flower color, it may be for fragrance, it may be for any like sterility is there, maybe for that, or maybe prickles or number of flowers. So, any kind of traits. So, here it is for the disease resistant like black spot resistance in roses, powdery mildew resistance in roses. So, likewise we can go for the selection of the plants, and then at the same time we can discard these plants which is having, which does not have the particular trait. So, in flower crops, what is the procedure for marker assisted selections? So, first of all it is selection of the parents, then development of breeding or mapping populations, then isolations of DNA from each plants, screening and then scoring of the molecular data, or you can say the genotyping, or then after that correlation of the molecular data or genotyping data with the phenotyping or morphological traits. So, first of all selection of parents. In case of flower crops, so first of all in case of flower crops it is very difficult to develop the mapping populations because these are the highly heterozygous crops. Like rose, it is a highly heterozygous crops, it is about the modern roses, that is Rosa hybrida, it is a cross between the 10 species. So, it is very difficult to develop the mapping populations. Okay. So, like chrysanthemum, chrysanthemum again it is a heterozygous crops, it is again it is a cross between the 6 species. So, it is very difficult to develop the mapping populations. So, whatever the varieties or germplasm we are having, we have to select the contrasting characters for that particular trait, or it should have a divergent origin, and we know that it should be homozygous. And then if it is a self pollinated crops, then we should have a pure lines, and if it is a cross pollinated crops, we should have a inbred lines. So, developing, yes.
What is the heterosis percentage in rose, because it is highly heterozygous? No?
Yeah, it is highly heterozygous. It is approximately 60 to 70% heterozygosity is there in roses, because it is a cross between the 10 wild species, the modern roses. So, it is very difficult to develop the populations. If we are also trying for developing the populations here, we are crossing the 2 varieties. Okay, we have a known varieties. Okay, we are crossing those varieties and we are getting the F1. When we are germinating the seeds of F1, again we are getting the segregation in F1 only because of the highly heterozygous material. If we are going for again selfing in that, and then again we are growing these plants, again we are getting the segregation. So, it is because of the
F2 means the segregation ratio is 9:3:1 or 3:1?
In case of
rose means it is highly heterozygous.
Yeah.
Then which pattern you get? F2 9:3:1 or 3:1?
No, actually in case of roses it is 3:1 we are getting the in F2 populations. But since it is highly heterozygous, so sometimes we do not get the such ratio. Okay. Since it is highly heterozygous, we do not have the these pure inbred lines in case of roses or in case of the chrysanthemum. Sometimes we do not get the particular ratio in F2, or even we are not able to develop the rills or nails because of its highly heterozygous nature in rose and chrysanthemum. And most of the ornamental crops, they are the vegetatively propagated crops, and since they are vegetatively propagated crops, they are highly heterozygous. If they are the seed propagated crops, then we can develop the mapping populations. So, in general, the mapping populations, these are the, like if it is a marigold crop, this is a cross pollinated crop, but there we can develop the mapping populations, and this is different kind of mapping populations are present in general. And seed propagated ornamental crops like antirrhinum, petunia, many seasonal flowers are there, antirrhinums like marigold, there we can develop these the populations like recombinant inbred lines, nails, F2 backcross, double haploids and F2 derived F3 populations, etc. So, this is the diagrammatic mapping population of self-pollinator species. This is in general, and this is the general genetic segregation ratio in different marker population combinations, where this dominant and co-dominant markers. If you are using the SSR markers, then we are F2 population, we are getting 1:2:1 ratio when the seed propagated crops, seed propagated ornamental crops. And this is the hypothetical gel photos which represent the segregating of the co-dominant markers and the dominant markers for the typical mapping populations for the F2, BC, backcross, rails and double haploids. This is how then we have to go for the sampling, and then PCR, lactophoresis, these are the procedures for it, and this is the overview for the marker genotyping. And again this is a marker assisted scheme for this early generation selection and a typical breeding program for the disease resistance. And this graph, it shows the advantage of the marker assisted selections over the conventional back crossing. Marker assisted breeding or marker assisted selection, it reduces the number of generations. So, this is for marker assisted breeding, where the generations they are reducing 1, 2 and 3, while in case of conventional back crossing methods, the more generations we have to wait for the go for the more generations. So, marker assisted selection, it is reducing the time as well as reducing the generations. So, there are some examples since flower crops using marker assisted selections, very few varieties were developed all over the world. In case of maybe in case of the roses, in roses again it is a heterozygous crops, and in case of the diploid roses, most of the work it was done in diploid roses, and in tetraploid roses or also it was used, the QTLs were developed for particular traits. So, this is the example marker assisted background selection for the integration of the black spot resistant into the cultivated roses or the commercial or modern roses. Okay. So, here they introgress the resistant gene, that is RDR1, which is the resistant gene to the black spot resistant. This is the dominant black spot resistant gene is a dominant gene, and they use the AFLP markers to develop the QTLs. So, here what they have used, they use the breeding schemes for the integration of black spot resistant from a resistant diploid line to the tetraploid rose varieties. What they have, so they have used the modified backcross strategies. Normally backcross breeding we used to do in the seed propagated crops, but since it is a vegetatively propagated crops and highly heterozygous, so they have used the modified backcross strategies to develop the use the marker assisted breeding to develop the varieties which is resistant to the black spot. So, they used this diploid rose, the line they were having, and they doubled the chromosomes they have developed, and then they develop the another, they develop the double the chromosomes using the colchicine treatments, then they got this line. So, they again this line they have crossed with the caramba, which is the susceptible line, which is a commercial variety, but it is susceptible to the black spot. And then after that they did the marker analysis using I think AFLP markers, and then they screen the plants, okay, which is having the characters from this CT54 line, and then again they develop this next line. So, they modify this strategy, they have not back crossed with the carom because it is susceptible, and they cross with the another variety. And then again they have used the AFLP markers for the marker analysis, and then they develop this another line. So, this line they develop, and because of the more inbreeding depression, so they develop this strategy. They can, because of highly heterozygous crops, cross pollinator crops, and they and more inbreeding depression is there. So, that is why they have modified this back cross breeding strategies. Okay. So, they selected as a recurrent parent in each generation to retain the vigor in the population.
Ma'am, this exchangeable, it is sometimes used male or sometimes used female. Why?
Where? Which one?
In second case you see.
Yeah.
Here the third case, here the tetraploid used as female.
Here.
But in this case, ma'am, it is used as female. Why?
This
Last one.
This there. Here also it is used as a female, and there also they have used as a female, and they have crossed with this also, and then again this also, and then again they have developed which is having resistant genes. They have developed these lines which is having the resistant gene, and they have a character of these varieties. Okay. Which cultivar? Yeah, actually they want the characters of this variety and resistance from this line, because this line is resistant to the black spot. So, they want the gene from this line, but they want the character from these other, because actually when जब उन्होंने इससे cross, when they have crossed with this line, so they got the plants or population which is having a character of this lines, and these lines have a characters of the wild species. And modern roses or hybrid roses, we do not want the characters of the wild species. Okay. Wild species means they have a very the flower size was very small, petals numbers were very less. So, that is why they have to retain its vigor, they have crossed with the this variety, but their purpose was to transfer the gene from this variety to the, and at the same time they have to retain the vigor, they have to retain the characters of these commercial varieties, those are susceptible to the black spot. So, that is why they have modified this back cross breeding strategy for the, and it is not only used for the roses, but they are also using for the other perennial crops like apple, because apples they are also the perennial crops, vegetatively propagated crops, and they are using this strategy to develop the varieties in apple which are resistant to the diseases, okay, like apple scab. So, here I am giving some of the examples that now they have developed the tightly linked markers to the to particular traits, like very recently they have developed these markers for the traits like mode of the blooming, recurrent versus once flowering, because recurrent flowering is very important in case of the roses. Generally in roses what happened, once it flowers, it gives the flowers which is having the commercial value. Then they give the second flush which is having the commercial value. But in case of roses, we need the recurrent flowering, so that it should start from like for example, it should start from the December here. So, January, Feb, March continuously it should be flowers, but in roses it happens first flush and then second flush, which is economically important. So, the recurrent it is very important, recurrent flowering is very important. Number of petals, because in wild species the petals are very less, 5 petals or maybe 10 petals, 20 petals in species, but in modern roses we want the more number of the petals. Okay. So, that is why this trait is very important. So, petal color, again we have a red color varieties, but now the demand is for the yellow color varieties, or another maybe the new combination of the color scheme. So, where the petal color is also very plays an important role. If we have attractive yellow colors, so definitely that will be an economic part. And then self incompatibility, because in roses self incompatibility is there, at the same time cross incompatibility is also there, which for that it is very important to find out that which marker is tightly linked to this particular trait. And fragrance, now modern roses, okay, modern roses or hybrid roses, they do not have the fragrance, and now the demand is for the fragrant roses, whether the fragrant roses for the cut flower purpose or for the garden display purpose or for loose flower purpose. So, again fragrance is again very important trait. And then vase life, in roses, the roses or any flowers, whether it is a chrysanthemum, carnation, the vase life is very less. Okay. So, that is why to enhance the vase life, or which marker is tightly linked to the vase life, it is very important to find out. So, they have find out this, they have find out for the vase life also. Disease resistance, like fusarium wilt in gladulus is very, it is a very important disease in gladulus, it causes a serious losses in the gladulus, like in powdery mildew and black spot dieback in roses. Okay. So, so that is why they have developed the disease resistant for powdery mildew in roses. And plant architecture in chrysanthemum. Okay. In chrysanthemum, plant architecture is very important, because in chrysanthemum spray chrysanthemums are there, standard chrysanthemums are there. Okay. Standard spray chrysanthemum, if they have a more number of the branches, so a single branch with the primary branch with the secondary branches, we can use it as a bouquet. So, plant architecture is very important in case of some chrysanthemum. And in roses, the QTL mapping was also done for another traits like thorn quality, growth figures, flower colors, number of flower petals, leaf size and prickle size. Prickles are very important in roses. Now, we know that the demand is for to develop the thornless roses, but when we are developing the thornless roses, at the same time they are also susceptible to the diseases. So, there the prickles rose plays a major role to develop the varieties which have a resistance to the diseases. So, here we are also working on this crop improvement program. For this, one of our students, she worked on this association analysis for the flower related trait in Indian bred roses. So, she developed the different populations, find out the different populations and other thing, and she find out the markers which are associated with the particular trait, like these markers. She found out of the 200 markers, she found that these are the markers, they are really associated with the flower diameters. Now, the next part is now validation of these markers, whether these markers they are really associated with the flower diameter. Similarly, she find out that two markers they are they are associated with the stem girth, internodal length, and then she got the one marker that is associated with the anthocyanin content, because anthocyanin content again it is the important trait in roses. And number of petals, one RH72, this marker is associated with these patterns. So, she has worked on these aspects. Now, the chrysanthemum. In chrysanthemum QTL mapping, it was done for the many traits like inflorescence, plant architecture trait, leaf traits, flowering time, aphid resistance in branching traits, water logging and this white rust and the drought tolerance also. But in chrysanthemum, the main problem lies in the slow process of marker assisted selection in the chrysanthemum. The population sizes and marker numbers are too small for accurate high resolution mapping, especially for the linkage analysis. The detected QTLs are mostly minor effect and environment specific QTLs with no validation, which are not robust enough for selecting phenotypes and performing positional gene clonings. These are the problems in the chrysanthemum to go for the marker assisted selection. So, it is very difficult to determine the location in the genome of these detected associations or candidate genes due to the lack of complete genome information. In case of flower crops, the genome size of the flower crops is more than other crops, vegetable crops or other field crops. Like if you take the example of chrysanthemum, its genome size is 9.4 giga base pair as compared to the other crops like in rose, and moreover the complete genome sequencing it was not completed in the most of the commercial flower crops. Then QTL mapping and BSA studies are currently based on the F1 populations. Since it is very difficult to develop the other mapping populations in all vegetatively propagated crops, so they are restricted to the F1 populations. So, the fruitful markers, they are really linked to the particular traits. So, it is very difficult to find out the making the linkage maps or find out the QTLs. So, in chrysanthemum leaf traits, they are also very important, because in chrysanthemum the base life of flowers it is more as compared to the leaf, because leaf they turned yellow before the flower senescence. So, the maximum leaf traits they are very important in case of the chrysanthemum. So, again another example is in case of Fusarium oxysporum in carnation, there they are also determine the mode of inheritance of resistance as well as to tag using random oligonucleotide primers the major gene involved in the manifesting resistant phenotypes. So, they have also this is the protocol how they have developed the varieties resistant breeding lines for particular this Fusarium oxysporum disease, and they use the RAPD markers for this marker designators. So, and they use the design sequence specific primers in efforts to develop the then SCAR markers. So, this is how they have used RAPD markers to develop the tightly linked markers. And then another example is in petunia, particularly for the crop timings and quality traits. Here they evaluated the diversity of two F2 interspecific hybrid petunia populations, and they used the they developed the SSR markers from the currently available Petunia axillaris expressed sequence tag sequences that will be useful for future genetic mapping and quantitative trait locus analysis. So, here they have used the F2 populations to develop the F2 hybrid mapping populations were developed by self pollinating of this Petunia axillaris and crossed with the Petunia exersta, F1 individuals, and by crossing Petunia integrifolia and exersa F1 individuals. Okay. And this is how they are showing the population distribution for these hybrids for the flower width and for flower height. So, this is for the cross between the Pusa excelsa and excelsa, and then integrifolia and excelsa B and C for the flower width and flower height. So, they have developed only the markers which are linked to the to that particular disease. And in case of marigold, they develop the markers which are linked with the male sterility in case of this marigold. So, they used these lines, male sterile lines, and crossed with the another inbred lines. Segregated populations of 167 plants constructed, and then they used the ISSR markers 38 and sequence related amplified polymorphism 170 techniques combined with the work segregant analysis to develop the markers which are linked to the traits. Because in case of marigold, male sterility is important trait. If we have a this male sterile line, genetic male sterile lines, then there is no need to go for the emasculations. So, it plays a very important role to develop the hybrid seed production. So, this is how they have used and developed the markers which is linked to the male sterile lines. And they found that this SCS 48 markers which is tightly linked to the TEMS genes, and it is responsible for the male sterility. So, since flower crops, very few work has been done on the marker assisted selections, and most of the work it was done out of the India, and in India most of the work it was done up to the molecular characterization, and then now it is we are going forward to develop the markers like in rose, which marker is associated with the which trait. So, there is in future there is need to develop the mapping populations in the seed propagated ornamental crops like petunia, antirrhinum, pansy, marigold, and at the same time association mapping it plays an important role in vegetatively propagated crops like rose, chrysanthemum, carnations. So, these important, and marker assisted selection it plays an important role over the conventional plant breedings. So, this is how we can go for the marker assisted selection in flower crops, but since it very few work it was done in the floriculture or flower crops. Why they have done because it is very difficult. These most of the floriculture crops or flower crops they are highly heterozygous. So, it is very difficult to maintain them, to propagate them, and then to develop the mapping populations. So, that is how less work it was done on the marker assisted selections. In roses, Mr. Dabner is there, he is working on the diploid as well as the tetraploid roses, and they have developed the up to the F2 populations, okay, for developing the like black spot resistant markers or markers linked to the powdery mildew resistance, etc. So, if you have any query regarding this, so you may ask the questions maybe from the floriculture students. Anybody from floriculture? You are from floriculture. Okay. Do you have any query about this? Yes.
Whereas marker resistant breeding is suitable for them, where it will come correct
accuracy level will be correct.
Accuracy level?
Accuracy level will be correct if you use a marker adjusted method in that polyploid species.
Polyploid species, yeah. So that's why actually most of the flower crops, they are the polyploid in nature. Rose is also polyploid in nature, chrysanthemum is also polyploid in nature. So that's why, and